Okay, so last time we left off right here, I believe, which is where we're extracting the different food molecules. This one's by aerobic organisms, so it's not necessarily humans, but yes, it also includes humans. Okay, and so you can actually break this into several stages. This is the typical, most common version of this, and it only accounts for three, which are the main three things in your diet. Okay. And those are lipids, polysaccharides, and proteins. And so you have stage one here, which is going to be taking the lipids, polysaccharide, and protein. You're okay with polysaccharide, right? Okay, fantastic. And the lipids are going to be broken down into fatty acids and glycerols. Remember, the glycerol is the head, right? And then you have the polysaccharides. These are going to be broken down into glucose is almost always in these figures separated out separately because it's so important. Okay? You actually have parts of your body, like your brain, your heart, etc., that only feed on glucose. They don't feed on anything else, even other sugars. You're screwed. Okay? Well, you're not screwed because you can infer it. And then the other sugars are less important, so they just get other sugars. This right here is then the proteins. They have to be converted into smaller molecules, which are the amino acids. Note that in all of these cases, you are converting them into something that can be put through cells and transported, okay? Because they actually have to be put through, transported to where they're going to go, and put into cells to do the processes, right? And then you have stage two. Stage two is kind of interesting because everybody thinks of them as completely separate things, but they're not. They all join together at the end. It's actually a hydra. Okay, see how it's a hydra? You have the body here, and they have the multiple heads. Okay. And so stage two is going to be getting down to the acetyl-CoA. Now there are actually other molecules that you could have called as the summary step here that are actually overlapping molecules that would go into citric acid cycle. But acetyl-CoA is by far and away the most important one, and this is what we actually named the citric acid cycle for, is a derivative of the acetyl-CoA that will be made during the citric acid cycle. And the acetyl-CoA is the easiest one to keep track of because of the CoA, coenzyme A, which is that adenosine on the coenzyme A. This is actually going to be fed into the citric acid cycle. You'll release the coenzyme A, which can then go pick up in another acetyl up here. And this is actually a cycle. So we'll recycle carbon atoms over and over and over in this. Okay? So you have the previous cycle going and some of their carbon atoms feeding into the new cycle. You don't actually use up the molecules completely. It's not an enzyme though, because it's different atoms every time you complete the cycles. Does that make sense? Like if you have enough cycles, you have none of the same atoms. You might have the same coenzyme. Only maybe. This is going to produce 2 CO2s. This is going to produce 8 electrons being transferred. These electrons being transferred are the energy source that is that. Remember how we were talking about what's going to generate the ATP is actually going to be a conductance across a membrane, right? That's that conductance. We're moving charge. This probably would have been easier to call protons in some ways, but it is more correct to call electrons because the electrons are what moves from here. Do not think of oxidative phosphorylation and the citric acid cycle as two different things. Okay? They're not. This oxidative phosphorylation, the step here is the step here. The step here, that is the step here. These are steps in this. They have the same enzymes that are involved. Same molecules are involved. We're just looking at different sides, different parts of it. Does that make sense? Okay. These are completely coupled. Dividing them like this is like taking your engine and your drivetrain and saying that your engine isn't connected to your drivetrain. And the teeth on the engine is different than the drivetrain. It doesn't make any damn sense, right? Yeah. But when you want to talk about, what the drivetrain is and what the engine is, you do, don't you? They're named different things. Okay. This is going to take oxygen, it'll convert it into water, which is that reduction of the oxygen here. This is going to give us ATP. And again, that's why the electrons are pointed out, because it's a reduction of the oxygen. We're looking at the oxygen here. Okay? Okay, that's stage 3. Now, the metabolic pathways contain many recurring motifs. You have the activated carriers. These are going to be small molecules to which a chemical group or electron have been added, which can then be donated to another molecule. I am recording, right? Yes, I am. Okay. These are going to frequently act as coenzymes or co-substrates. Again, coenzyme meaning something that comes along with the enzyme for it to do its activities. Typically, it was not going to get consumed and used up, like coenzyme A. Co-substrates, these are going to be substrates that have to come along with another substrate, right? Okay, remember that we oftentimes are going to skip those co-substrates as being the primary substrate, and that's because we're following along a train, right? Yeah. Okay, so an example is going to be the ATP is going to be an activated carrier of the phosphoryl groups. And so ATP's function is actually to transport the phosphoryl groups, okay? The ATP itself is actually kind of meaningless, which is why we oftentimes go AMP, ADP, ATP, because we're actually more interested in how many phosphoryl groups are. The fuel molecules are going to transfer electrons to the carriers, which then transfer their high potential electrons to oxygen. This is the oxygen you breathe in, and you did all that work carrying around, right? Your hemoglobins. The nicotinamide adenine dinucleotide. This is NAD+. I know, have you ever seen the full name of that before? Yeah, probably not. Yeah, the biology, they never say the full name, but they just use NAD+, NADH. Yeah, never explain what it is. Anyways, that's what it is. This is going to accept a proton and 2 electrons in the oxidation of a substrate to form NADH. The reactive part is actually its nicodinamide ring, which is what we're talking about here. Notice again, this right here is the part we're talking about oxidizing. You see that? So we're oxidizing this to a carbonyl. The NAD plus is going to NADH. Again, this is your reactive site right here. Hey. It's okay. It's okay. I went ahead and got started without you, but you can check out the video. And sorry. That's fair. That's fair. Again, the video. Okay. No problem. You missed the big reveal. NAD plus had its full name said. Hey, no, you've had all these classes with NAD plus and you never knew what the hell it was. There you go. It's like your favorite superhero being unmasked. Okay, so your flavin adenine dinucleotide. Notice the names here: adenine dinucleotide, adenine dinucleotide, adenine being the A and ATP, right? Or adenosine, technically. These are derivatives of each other. So the ATP is derivated to form your NAD and your FAD. Okay? And the AD parts are the same thing for both of these. So anyways, flavin is the difference there. This is going to accept 2 protons and 2 electrons in the oxidation of a substrate to form the FADH2. The reactive part is going to be the isoalexene ring. Excuse me. And again, this is going to be your reactive sites here. Okay? Okay. And so notice that was your reactive site before, and these are heterogeneous conjugated rings. What did you guys learn about heterogeneous conjugated rings? They have these like electron pairs that you can put stuff on, and then they can share charge around that ring. And so they're very good at like losing and bringing these things on because they're pretty stable both ways, right? Ooh. So that's why they make really good electron transfer. Because they can stabilize that. And this is an example of a oxidation being performed with FAD being the agent transporting the hydrogens there, being the other side of that. Remember, anytime you have a redox reaction, an oxidation or reduction, you have to have the other one as well, don't you? Okay. Okay, and so this is the isoalloxene ring. It's going to be a derivative of your vitamin, which is riboflavin. You may have noticed, by the way, flavin, as in the flavin from riboflavin, right? Okay, and so this is actually the oxidized form of FAD right here. And you notice, why is it the oxidized form? What if we not have on it? Hydrogens. So you may have noticed, I remember I was like, one of the ways of keeping track of what's the oxidized and reduced form in biochemistry is to look at the number of hydrogens on it, right? If you gain hydrogens, you've been reduced. If you lose hydrogens, you've been oxidized, okay? You can also look at the oxygens. If you gain oxygens, you've been oxidized. You lose oxygens, you're reduced, okay? Following the electrons is much harder. The oxidation numbers in electrons is a lot harder in biochemistry because these things are very large molecules, right? Compared to like Gen Chem. I know, right? Like 5 atom molecules, and then we're going to like 50 atom molecules minimum, right? Yeah. So in most biosyntheses, the precursors are going to be more oxidized than your products. The ATP and reducing power are going to be needed. An example is going to be 4 electrons are needed to reduce the keto group to a methylene group. Again, this is your keto group right here. This is your methylene. Methylene being that CH2 repeated units that we had talked about back in organic. And again, if you look here, you can notice that we were oxygen bonds. They went to hydrogen bonds. And so we were reduced right here, weren't we? Okay. This water is being produced. The NADPH, is this going to be the electron donor in most reductive biosyntheses? You notice that we have the NAD portion right here. And now we have a phosphoryl pH, right? Okay. So the coenzyme A, we're starting into the sort of citric acid cycle stuff that's going to set us up. Okay? So coenzyme A, CoA, is going to be a carrier of acyl groups. This is going to be derived from vitamin B5, which is pantothenate. The reactive part is actually the terminal sulfhydryl group. Remember those right there? Okay. And so, do you remember your carboxylic acid derivatives? And we were talking about the thiol versions. And I mentioned to everybody that took my class that this is like a really nice moderate group that's quite reactive, and you can put stuff off and on really easily. Okay? And guess what? That's what it's getting used for, isn't it? So you can put these acyl groups onto this sulfur here fairly easily, and then you can break them back off fairly easily. Okay? Okay, so the acyl groups are going to be linked to CoA. This is again via thioester bonds that are going to form the acyl-CoA. Again, this is the acyl group right here. This has an R, so an indeterminate number of carbons, right? This right here is the acetyl group, or acetyl group, okay, depending on which side of the pond you're on. This is going to be with 1 CH3. Right. So the acetyl that's linked to CoA is again going to be the acetyl-CoA. Everybody comfortable with that? Okay. Is this right here part of the CoA or part of the acetyl that's going to get used up? The sulfur is part of the CoA. Okay? And it's the thing you're going to link to. The carbonyl is part of the acetyl. All right, so this right here is the coenzyme A. What do you notice about this part right here? The green. A, D, P. A, and then ribose right here, though it is phosphorylated, and then 2 P's, right? Yeah. And right here, this is the pantothenate unit right there. And then this is the beta-mercaptoethylamine unit. And this is where you actually get your thiol group right there. That is going to be where you attach and detach these acetyl groups. Okay? Everybody okay with that? Fantastic. Okay. So your transfer of the acyl group is going to be an exergonic. This is because the thioester is thermodynamically unstable. And that's why I was mentioning that thioester is very good for linking and unlinking stuff very easily. Okay? That delta G naught prime for the hydrolysis of the acetyl-CoA has a large negative value. And so what does that mean about this reaction of having the acetyl on the CoA? What's going to happen if you just leave that around in water? That is thermodynamically unstable, isn't it? Yeah. Okay, so the electrons of the CO bond cannot form resonance structures with the CS bond that are stable, as those cannot form from the CO bond. So right here, this is a normal ester, right? So normally we would have our carbonyl next to our oxygen. We can form a resonance structure, can't we? And this can actually share those charges, and it makes this more stable. Okay? This right here, when we have the oxygen's ester, it's more stable because of that resonance charge. But the thioesters, on the other hand, they can't do that. And that's because this is unstable. The electronegativity of oxygen is significantly higher than sulfur, right? And so this is essentially mostly not going to form, okay? Meaning you really don't have a resonance structure in these cases. Right? Okay, which makes this much less stable than if you had something like that. Again, we're in another situation where we're depending on the thermodynamic instability, right? We're setting a rock up on a hill, and then we want to, when we want it to happen, we want to sort of give it a little prompting, right? And so we use the kinetic stability, the kinetic inability to go forward in the reaction to keep things from happening, whereas we're setting them up for being thermodynamically unstable, right? So that when they happen, when we give something to keep them going fast, like an enzyme, they can go along spontaneously, can't they? So again, kinetic stability is going to allow the enzymatic control over the flow of energy. The NADH, NADPH, and FADH2, these are going to react slowly with oxygen in the absence of a catalyst. If you just left them in the environment with oxygen, they will slowly oxidize over time. You'd be left with nothing. Okay? ATP and acetyl-CoA are going to hydrolyze slowly in the absence of a catalyst. And again, we're looking at running a race against time, right? Same thing as the fuel in your car. We have very high energy fuel. Why doesn't it burst in flames? Kinetic stability, right? That's what we're depending on. We're doing the same thing in our body, okay? Kinetic stability is keeping this from getting used up. It is thermodynamically unstable, so it can push stuff through. A small set of carriers is going to accomplish the majority of the exchanges of the activated groups in the metabolic pathways. It actually narrows down a lot. So these are some of the different activated carriers in metabolism. You have ATP. The group that's carried by ATP is the phosphoryl group. Hopefully you all kind of knew that, but you probably didn't formally recognize this, did you? Yeah. I mean, isn't that a nice formal recognition that you can do? It narrows everything down to a fairly small list, doesn't it? The NADH and NADPH, these are very similar, by the way. They're going to be carrying electrons. This is actually your vitamin precursor. Notice these vitamins are some of the most important vitamins you need to have in your diet. Guess what? Because without these, you can't carry the molecule, right? And then you can't do all the other reactions that everything depends on. This is the FADH2. This is going to carry electrons again. FMNH2. This is again carrying electrons. Coenzyme A carries acyl groups. Lipoamide carries acyl groups. Thiamine phosphate, this is going to carry aldehydes. Biotin is going to carry CO2s. You guys may have heard of this as being a key component in diets these days. This has had sort of a revolution. Before that was NADH was having a revolution. Anybody know why that stopped being a thing, where NADH supplements stopped being a thing? Apparently, if you have cancer, it makes the cancer just supercharge. Yeah. And so people that take heavy amounts of NADH2 supplements, if they get cancer, it's like they were didn't get discovered for a month or two more. It can just progress cancer super quickly. So very problematic. God knows what biotin does that we're not realizing, right? Tetrahydrofolate, this is going to be 1 carbon units that are carried. The S-adenosyl methionine, these are methyl groups that are carried. Uridine diphosphate glucose, this is glucose that are carried. You'll actually see this later on when we get into other metabolic pathways. Cytidine diphosphate diacylglycerol, these are phosphadates that are carried. Nucleoside triphosphate, guess what they carry? Nucleosides. That's that DNA and RNA synthesis that we were doing, wasn't it? Ooh. Wrapping back around. So again, the vitamins are going to be organic molecules that are needed in small amounts in the diets of some higher animals. Higher animals is probably a bad terminology now, but you get the idea, right? Things that can go out and hunt stuff. Okay. That can, move around and get different plants and get different things in their diets. These must be modified to serve its function. Most activated carriers that act as coenzymes are derived from these vitamins. This is why you need a fairly limited number of them, because the carriers don't get used up all the time. You just need to replace them every now and then. Okay? But on the other hand, they're absolutely vital for you surviving, right? Okay. And so you absolutely have to get those in your diet. So these are the B vitamins, the thiamine, B1, you get the idea. Does anybody actually need me to go over this? Okay. Yeah, you can go over that in your own time sometime. These are what those look like. Notice that was the riboflavin right there that we had previously talked about. These are the non-coenzyme vitamins. These are the vitamins A, C, D, E, and K. These are actually their functions. And this is what their deficiency would cause. Anybody have any idea what the most common disease, the third most common disease on college campuses is? Okay, when you guys get out from under your parents, a lot of you may end up just eating pizza and stuff all the time, right? Or Chick-fil-A. And what do you not get in your diet? Fresh fruits. Yeah, fresh fruits. And so you don't get vitamin C in your diet, and this can cause you to contract scurvy. So, yes, scurvy like a pirate. It's also very common in college students. So, pirates and college students. Scurvy. The key reactions are going to be reiterated throughout the metabolism. Thousands of metabolic reactions can actually be subdivided into 6 types because of this. Because you may have noticed that when we had up in this table here, there was actually a very limited number of types of things, wasn't there? Okay? And this causes all metabolic reactions to collapse down into these 6 types. There's the oxidation reduction reactions. This is going to be some form of electron transfer, right? Though typically not as electrons being transferred, typically as hydrogens or oxygens. Okay? Group transfers, these are going to be transfers of a functional group from one molecule to another. There's hydrolytic, these are going to be cleavage of bonds by the addition of water. Carbon bond cleavage, this is going to be means by other than hydrolysis or oxidation, because those were covered here, right? And so it's the other stuff that doesn't fit in those. Okay? And so 2 substrates are going to yield 1 product or vice versa. Remember that when we look at these reactions in a metabolic sense, both directions are actually equally valid, right? Do you need to make ATP all the time and nothing else? No, sometimes you just want the damn adenosine, right? You don't want the triphosphate portion. Okay. Isomerization, this is going to be a rearrangement of atoms to form isomers. Everybody remembers isomers? Different arrangements, same overall chemical formula. Okay. Ligation, this is going to require ATP cleavage. This is going to be the formation of covalent bonds, carbon-carbon bonds. Anybody ever heard of the term to litigate? Nobody has ever heard of I'm having litigation done? No? It's what lawyers do to you. It's the same root word, in case you're ever wondering. Yeah, ligation, litigate. Yeah. And yeah, it means the same thing to join in a contract. Yeah, to join. So the oxidation reduction reactions is going to be useful energy is going to often derive from the oxidation of carbon compounds. This is the primary energy source of most aerobic organisms. I don't think there is any aerobic organisms where it's not actually, but I'm getting out ahead of myself. I'm future-proofing me. In 40 years, when they find an organism that's aerobic and can do this without carbon atoms, Dr. Clausen, he was careful, okay? You have to think about that. Okay, so some of the examples are going to be the oxidation reductions of the citric acid cycle, of course. Here we have succinate with FAD going to fumarate. By the way, a big hint. Oxidation and reduction reactions are going to involve the transfer of what? Electrons. Transfer of electrons typically are done by what groups? FAD, NAD+, NADPH, right? If you see those things, very high chance. Okay? Very high chance. This is the group transfer reactions, so it's going to be used to synthesize ATP and in signaling pathways. We've actually seen a number of examples of this already, in like the phosphoryl groups transferring, etc., right? So some of the different group examples are going to be phosphoryl group transfers, of course, which I just mentioned. Let me just repeat myself a bunch of times. Glucose is getting a phosphate from ATP, right? Right there. Okay? And so, yeah, you can have kinases doing this, etc. Hydrolytic reactions, it's exactly what you think it is, cleaving things with water, right? Okay, so hydrolysis is going to cleave bonds by the addition of water, commonly used to degrade large molecules. You can have, of course, the hydrolytic cleavage of proteins, which we've actually seen a number of times before, right? And so that was hydrolytic cleavage. By the way, is metabolism just the processes involved in eating things? No, it's the breaking apart and making of all the molecules in your body, essentially, right? Yeah. It's a very common misconception. People, metabolism, and they just think about the eating things. Yeah, I mean, all your molecules, or at least the vast majority, come from eating things, but you also have the stuff you're recycling. Okay, so carbon bond cleavage. This one's going to need 2 slides to deal with because they can be kind of big. This can occur by means other than hydrolysis and oxidation, of course. Here we actually see an example where it's not hydrolysis, it's not a redox reaction. So the conversion of the 6-carbon molecule fructose, 1,6-biphosphate, which is this one right here, into the 3-carbon fragments. This is during glycolysis. We haven't actually gotten to this full reaction pathways for these, but this is one step from that. And that's that right there. And as you can see, that you haven't really oxidized or reduced anything. You haven't put water across this, have you? So you need another category for this. The dehydration is also an important subclass to this. So some of the example of that is the generation of phosphoenol pyruvate from 2-phosphoglycerate. So here's your 2-phosphoglycerate. We're removing water, forming a double bond. Okay? That was that dehydration reaction. All right, isomerization reactions. A big hint to this is typically they end up with an iso on one side of it. Because isomerization, iso. This is going to be a rearrangement of the atoms within a molecule, typically to prepare the molecule for subsequent reactions. Basically, you're just setting up the next one almost all the time with these. Some of the examples will be the conversion, of course, of citrate to isocitrate. And so here we have citrate, we rearrange this, and we get isocitrate. Ligation reactions is going to form bonds using free energy from ATP hydrolysis. One of the examples is the formation of oxaloacetate from pyruvate and CO2. Oh, I lost my reaction. Shit. You can imagine, okay? You're forming bonds. We've actually seen this a number of times at this point, where we used ATP energy to form a bond and make something by forming those bonds, joining two things together, okay? Yeah, sorry. You don't get a nice example for that one, I guess. Yep. Metabolic processes are going to be regulated in three principal ways. If somebody wants to send me an email, that would actually be very helpful if you get a chance, because I'm probably going to forget by the time I'm done with lab and all that. The metabolic pathways must be regulated to create homeostasis. This is a stable biochemical environment. Metabolism is regulated through these three mechanisms: altering the amount of enzymes, restricting the accessibility of substrates, and regulating the catalytic activity of enzymes internally or externally. Notice, by the way, that we've actually built ourselves up to a point where when I say those three terms now, did anybody get lost? No. We've actually seen all those, haven't we? Yeah. So, I know, right? Looking back, you actually have learned something. Why does this create a stable biochemical environment? What if we just used up something over and over and over and over? What would happen if we never replenish something? Yeah, well, it would run out, but we'd also have another problem, which is, what are almost all of these reactions? They're equilibria, right? If we just use it, and we build up the other side of it over and over and over, it's going to start pushing the other direction, isn't it? So we have to do both. We have to replenish and remove, right? And it's that regulation and that complex series of creating a homeostasis environment where we're bringing in or removing things to maintain some sort of balance. This is what creates our ability to live, okay? So controlling the amounts of enzymes. The amount of a particular enzyme is going to depend on both its rate of synthesis and its rate of degradation. These are two of the main factors that are going to be involved. The level of many enzymes is going to be adjusted by a change in the rate of transcription of your genes and coding them. We've actually seen other parts that act on this, but I'm giving you this part. And I'm going to be honest, like, about this time, we're going to start doing this thing where we're going to come back into stuff we previously learned and layer on some complexity onto it, okay? So if you're wondering why we're like recovering stuff, that's why that happens. Anyways, so compartmentalization is also going to often segregate opposed reactions. Some of the examples of that is the fatty acid oxidation. This is going to occur in the mitochondrial matrix, while the fatty acid synthesis, this occurs in the cytoplasm. Why would you want to segregate these things? Okay, let's imagine a human that's just one giant compartment, okay? That's all they have. So all reactions happen in the same space, okay? What would happen whenever you're trying to form more glucose in one area and you're trying to form the starch or cellulose in the other area? Would you be able to do it? No, because everything would affect your equilibriums, right? Yeah. And so it's incredibly important to compartmentalize your reactions. And because we have base units, which are cells, which mostly have to survive and do their stuff mostly on their own and can't completely depend on something else, we need subdivisions and compartments within those cells, right? And that's why we have things like the mitochondria that still exist and didn't just, get forced out and go off and die. Okay? So another thing is controlling the catalytic activity. Does everybody understand how controlling accessibility of the substrates changes your ability to have reactions go one direction or another? Okay. So like if you bring in more reactants, you shift the reaction towards products, don't you? And then you can remove the products from that compartment, and that means you have a locally elevated concentration of substrate that'll push the reaction through, won't it? Okay. Controlling catalytic activity. Catalytic activity is regulated allosterically or by covalent modification. Feedback inhibition is an example of the allosteric regulation. We have spent a few chapters doing that before. Concentration of the allosteric activators and inhibitors can be changed. We actually looked at that a bit in the different signaling pathways, didn't we? Where we were changing the activators and inhibitors in some of these processes. Reversible covalent modification control catalytic rates of enzymes. Again, we've covered all of these individually now. Now we're stitching them together into a full understanding. Okay. So the energy change, this is proportional to the mole fraction of ATP plus half the mole fraction of ADP. Remember, ADP is only going to count for a portion of ATP. So the energy charge here, charge, is going to equal to ATP plus 1/2 the ADP. This is going to be over all the energy possible, which is the ATP, ADP, and AMP, i.e., all three forms of the AP. Okay? Why is AMP not on the top here? When we're using energy to move a reaction forward on something, do we normally cut off the monophosphate portion and produce P? No, we don't normally do that, do we? Technically you can, but it's not done to provide energy for reactions. It would be way more expensive to remake it than it would be to add more phosphates. So this right here is the last one that actually would produce energy for you. Do you remember your energy differences here? So this right here was like half the energy from going all the way from ATP down to AMP, as it was only 1.5 times the energy, wasn't it? That's what you're seeing. You're seeing 1.5, aren't you? Yeah. Okay, so again, this is going to range from 0, which is all AMP, to 1, which is all ATP. This is always going to be a fraction, 0 to 1. The ATP generating pathways are going to be inhibited, and ATP utilizing pathways are going to be stimulated under conditions of high energy charge. The energy charge is maintained within narrow limits. Typically, it's maintained in your cells at 0.9 to 0.95. The enzyme regulating pathways are going to be allosterically inhibited or activated by binding to the ATP or AMP. What is the ATP going to do? It's going to halt production of additional ATP, right? If you have AMP, what's that going to do? It's going to activate the production of additional ATP. Why would that do that? What's the form that has no energy for you to use? AMP. So when your ATP is very high, you have a 0.95, 0.9 sort of things. So you have a 0.95. That's like your battery saying 100% charge. And so your cell goes, I can disconnect it from the charger, I'm good. When you get down to a 0.9, that would actually be your battery being on that 20%. You know how it shuts off at 20% so it doesn't damage? Your cells do a bit of the same thing. So when you get down to only 0.9, it's going to go, oh, gotta make some more. We're in trouble. Get that on the charger. Okay, so the energy charge is going to be a useful concept for understanding how metabolism is regulated. This is again going to be your relative right here. This is your energy charge. And what we can see here is that your ATP generating pathways, you're going to contrail pretty rapidly when we're starting to reach 1, okay? But they actually pick up pretty rapidly once you get down past 0.9-ish, okay? And by the time you're at a 0.75, we're starting to get back in the linear ranges, right? And so you're actually at that point almost maxing out because you're really going fast if you get dipped down to 0.75. That's actually really low, okay? That'd be like your battery being on 30%. I should probably charge it, right? Ooh, I'm gonna be in trouble. This right here is your ATP utilizing pathways, and again, what we see is they actually pick up when they're at around 0.9-ish, right? And they get drastically higher, okay? And then they curtail off pretty quickly once we get down to the 0.75s and stuff, because that's very low, actually. Okay, anybody have any questions about that? All right. I'm actually going to take a little break for a couple minutes.